This tutorial will describe how to work with sequence rendering in Lead Discovery ChemCharts. To access sequence rendering options, right-click inside a ChemCharts table and click on the Property Dialog icon. From the Property Dialog, table characteristics from colors and fonts to column headings and sorting can be adjusted. In the Data tab, the table you started from will be listed. For this tutorial, we will focus on column renderers and columns that contain protein or nucleotide sequences. First, select a column that contains sequence information. Sequences may be present simply as protein or nucleotide sequences, aligned sequences, or Genbank columns which contain additional annotation information. Here I'll start with a column that just contains protein sequences. Different renderers are available for the various types of content that may be in the column. For this protein sequence column, I'll select the sequence renderer. There are various color schemes available to choose from, as well as a plain uncolored option. Some options are specific for proteins and some are specific for nucleic acids. Refer to the user's guide for details about the different color schemes. Sequences can be displayed in the table in three ways. Fit will display the entire sequence in a window to give a high-level overview. Scroll displays the sequence on one line and provides a scroll bar at this column header. And Wrap displays the entire sequence as multiple lines using the table's font style and size. For columns with aligned sequences, a comparison color scheme and a comparison character scheme can be chosen to illustrate the differences between the sequences. Also, if the sequence is a nucleotide sequence, it can be translated to protein sequences. I'll cover these options a little bit later. Individual residues can be displayed as single characters or using three-letter codes. Also, coloring can be applied to the characters of the sequences themselves rather than the backgrounds of the characters. To illustrate specific motifs of interest, a custom motif coloring can be added. Click on Add, specify the sequence of interest, and select a color. Multiple motifs can be added, but they should be non-overlapping, otherwise one color will overwrite another. To delete the custom motif coloring, click on the Delete icon. When a column contains aligned sequences, most of the options available remain the same, but there are also a few additional options. The top sequence of the pair is the reference sequence, and it is colored by the selected color scheme. The comparator sequence on the bottom can be colored also using the selected color scheme by selecting None. The Blossom comparison scheme colors the comparator sequence with different intensities of blue to red based on the level of similarity with the reference sequence. The Simple comparison scheme colors the background red for residues that differ from the reference sequence. The Simple character scheme will display a dot for any amino acid in the comparator that is the same as the reference sequence. Comparison schemes can also be applied across rows. In this example, I have a column of sequences that have been aligned using the multiple sequence alignment data function, and I would like to see the conservation across those sequences. If I lock or fix a row by double-clicking on the row header or right-clicking and choosing the Fix icon, the sequence of that row becomes the reference sequence that all others are compared to. A new row is inserted at the top to display the reference sequence, and the other rows are shifted down. The reference sequence is now responsive to changes in color scheme. All other sequences in the table are responsive to changes in comparison schemes. When a column contains Genbank data, again, most options behave the same. In this case, sequences and annotations can be turned on or off. Nucleotide sequences also have most of the same options that we've already covered for protein sequences. DNA or RNA can be translated into protein sequences, which are displayed below the nucleotide sequence. Options to specify the nucleotide type, the direction of translation, and the reading frame are provided, as well as a custom color scheme for the translated protein sequence. There are a lot of options to choose from for sequence rendering, and the best way to become familiar with them is to explore the column renderer's properties, trying out the various options. You can't modify the table's data content or inadvertently delete any data, and any changes can be easily reversed. Thanks for watching, and check out the other Lead Discovery ChemCharts tutorials.